Right, starting on with the next chapter on a relationship among organisms and evidences of evolution. All living organisms have arisen through the evolutionary process and shows diversity. All the microbes, plants, and animals show a great diversity. These shows a number of life processes which are common to all organisms, like all are formed of living protoplasm, derive energy and matter from the environment, maintain homeostasis. I repeat that, homeostasis through a number of metabolic processes. Evolution is the formation of newer types of organisms from the pre-existing ones through modification. Evolution is therefore called descent with modification, Darwin, 1859. I repeat, evolution is therefore often called descent with modification. Evolution has occurred through mutation, natural causes, selection, and isolation. Term evolution was coined by Herbert Spencer and has been derived from the Latin word evolver, which means untold and literally means the process of continuous change. Evolution is an orderly change from one condition to another. Example, when planets and stars change, and between their birth and death, then it is called a stellar evolution. Where matters, elements change from time, it is called as inorganic evolution. Evolution is of several kinds, like progressive evolution, retrogressive evolution, and microevolution. In progressive evolution, we talk of formation of more complex, elaborate, and specialized structures from simpler ones. Retrogressive evolution deals with the development of simpler and less elaborate forms from complex ones, and microevolution is development of minute changes due to gene mutations. And before we go into the connected details on the next page, let me just read out this historical aspect of evolution too. Those who would like to be aware of the ancient thoughts. According to Indian mythology, Brahma created various forms of life in one stroke. According to Anaximander, a great philosopher of the 6th century BC, water was the material source for evolution. Heraclitus, 510 BC, proposed the idea of struggle among living organisms. Plato, from 428 to 348 BC, proposed that each species has an unchanging ideal form eidos in his idealistic concept. According to him, all earthly representatives are imperfect imitations of such true essence of an ideal unseen world. God is perfect, and everything that existed on earth was his idea. Aristotle, in 384 to 332, constructed a scalar naturae, also called ladder of nature, a great chain of being in which he grouped the living organisms from simpler ones to the most complex ones. So he proposed a gradual transition from imperfect to perfect. The ancient Indian texts of philosophy and Ayurveda deal with the origin of life. Manu's texts in Sanskrit, Manu Samhita or Manu Smriti, 280, also made a mention about evolution. And the recombinations, a continuation from the first page. The change occurs The changes occur below the level of species. To specifically mention macroevolution, development of large changes like formation of new species and generation due to mutation, causing large-scale changes in chromosomes, right? And mega-evolution, large change given rise to new families, orders, classes. Parallel evolution, independent formation of similar traits by related groups of organisms. Convergent evolution, adaptive convergence, formation of similar traits by unrelated groups of organisms as wings in insects, birds and bats. Divergent evolution, or adaptive radiation, which is the formation of different structures from a common ancestral form, example forelimbs in horse, bat and human beings. Coevolution, which relates to evolutionary changes in one or more species in response to changes in other species of the same community. There are many types of evidences which support the process of organic evolution. These evidences are anatomical or morphological, embryological, paleontological, physiological or biochemical, cytological, molecular, biogeographical, taxonomic, and from genetics. Animal breeding, behavior, hosts and parasites, formation of new species, and experimental production of new species. Study of functional anatomy is known as lectology. Let's just get that clear. To make it clear, it's tectology, right? Anatomical or morphological evidences. Comparative study of the morphology and anatomy of groups of animals or plants shows that certain structural features are basically similar. These features include body organization and gradual modification. Morphological and anatomical evidences include homologous and analogous organs, connecting links, vestigial organs, and atavisms. Organs have similar embryonic origin and construction, but differing in external form and function are called as homologous organs. Homologous organs are found in forms showing adaptive radiation from a common ancestor, so these give evidence of divergent evolution. The vertebrate forelimbs, insect legs, insect mouth, parts, vertebrae, epidermal derivatives, tail and vertebral column, and thorns of bougainvillea and tendrils of passiflora are some examples, common examples of homologous organs. The forelimbs of these animals, seal, bird, bat, horse, and man, have the same general function of locomotion but have different shapes and bring about different types of locomotion. They are used for swimming in seal, flying in bird and bat, running in horse, and grasping in man. Legs of all insects consist of 5 pairs, named as coxa, trochanter, femur, tibia, and 1 to 5 joints tarsus, and are used for locomotion. They all have different form and perform different specific functions in different insects. The same basic structure of the leg shows that all the insects have a common ancestor. Variation is due to adaptive modifications during the evolution of the group. Vertebrate tail is composed of vertebral column, blood vessels, nerves, muscles, and skin. It lacks coelom and viscera. The tail is used for swimming in fish, mud puppy, amphibian, crocodile, and whale, for holding on to objects in seahorse, chameleon, scaly anteater, and New World monkeys. As a balancing organ in kangaroo, to keep off flies in cattle, and for defense in spiny-tailed lizard and wall lizard. Homologies are of 2 types. Phylogenetic, example the hand of man and leg of horse, and serial homologies in vertebrates. Analogous organs are those organs which have the similar function and are superficially appear similar and are different in fundamental structure and embryonic origin. Examples of analogous organs are wings of insects, birds, and bats, meant for flying. Phylloclades, cladodes, and phyllodes perform the function of leaf as photosynthesis. The analogous organs are the results of convergent or parallel evolution in separate lineage. There are different modified roots. Stems and leaves. Sweet potato is an underground tuberous root and potato is an underground stem, but the modifications are meant for storage of food. These are therefore an example of analogous organs. The organisms which possess the characters of two different groups are called connecting links and shows the possible path of evolution. Connecting links are the living intermediate forms. Okay, on to the next page. We come over to a table of important connecting links, right? We have the chimera, a rabbit or ratfish, between cartilaginous and bony fishes. Virus, between non-living and living. Euglena, between plants and animals. Proterospongia, between protozoa and porifera. Neopilina, between mollusca and annelida. Peripatus, between annelida and arthropoda. Balaeoglossus, between echinoderms and chordates. Lungfish protopterus, between fishes and amphibia. Latimeria, between fishes and amphibia. Seymoria, between amphibia and reptiles. Archaeopteryx, between reptiles and birds. Spaniantida echidna, between reptilia and mammalia. Duckbill platypus, between reptilia and mammalia. Lycohaps, I hope I pronounced that right, between reptilia and mammalia. Right, and the missing link are the transitional forms between two groups of organisms occurring only in the fossil state, example Archaeopteryx. Vestigial organs are the useless and functionless degenerate structures which were large and functional in some of the animals. They are rudimentary, relatively smaller in size and do not function. Right, and some of the vestigial organs in animals are given as follows: the vermiform appendix, coccyx, plica semilunaris, pinna muscles, and wisdom teeth in man, rudiments of hind limbs in python and boa, pelvic girdle in whales, splint bones, vestigious second or fourth digit of fore and hind limb of horse, wings of kiwi, and eyes in cave animals. Atavism is a sudden reappearance of those ancestral characteristics in an organism or in the organism of a group which did not occur normally or which represent the reminiscent of normal structures possessed by the individuals of other groups. Atavism is also known as reversion. Examples of atavism are cervical fistula. A man, only one pharyngeal pouch perforates to form an opening from the pharynx to the exterior in the form of the external ear canal and eustachian tube, but rarely neck may possess an additional opening representing the cervical fistula. Tail is absent in man, but occasionally a human baby is born with short, fleshy tail. Hairs on the body and face. Homodont dentition in crustaceans. Crustaceans, have I got it right? Cetaceans are mammals with heterodont dentition, but pecivorous cetaceans possess simple conical homodont teeth. Their ancestors are known to possess heterodont teeth of mammals. This is true reversal of evolution. The principle atavism was proposed by L. Dollo and is called Dollo's law. It states that living organisms do exhibit evolutionary irreversibility. Types of atavism are family atavism, race atavism, and atavism of teratology. Family atavism includes the transmission of individual characteristics or characteristics within the members of a family in latent condition for several generations and their sudden reappearance in the offsprings after a number of generations. This phenomenon is controlled at gene level and can be explained by simple Mendelian laws of inheritance. Race atavism includes those cases of reversal where one or more characters of one race appear in the individual of another race. Example, profuse growth of hair on the body and face of Irish dogmen, etc. Atavism of teratology includes the appearance in arrays of such abnormal characters which were normal in other supposedly ancestral races. For example, appearances of cervical fistula in man which actually corresponds to gill slits, etc. Evidence from embryology. Embryological development reveals the unity of plant embryos of the vertebrae. CDs exhibit many features that are absent in the adults. For example, all the embryos, vertebrates, including human, develops a row of vestigial gill slits just behind the head. Right, we're into the next page, and we have another table. Evidences from embryology, which shows relationships again. Similarity in early development. Resemblance among vertebrate embryos in shape and structure. Recapitulation theory of biogenetic law by Ernst Haeckel, stating that an animal in its development from egg to adult repeats or recapitulates in a condensed form the stages through which its ancestors have passed in the course of evolution. That is ontogeny and repeats phylogeny. Temporary non-functional embryonic structures, retrogressive metamorphosis, and neoteny. And we have, on the other hand, larvae of Herodemania shows characters of chordates, presence of notochord, change of a fish-like embryo of human to a 7th-month human fetus resembling a baby age to a human baby, origin of gymnosperms from pteridophyte-like ancestor, fish-like tadpole or frog shows it has evolved from fish-like ancestor. The protonema of moss and fern shows an algal ancestry. The presence of primordia of leg in each segment of the embryo of insect shows that they arose from an ancestor having segmentally arranged appendages. All right. We proceed on to the latter aspect. Evidences for embryology are used on the comparative study of the embryos of various animals, which is shown in the flowchart above. In 1828, von Baer put forward Baer law. E. Haeckel law called it the biogenetic law in 1868. The view was summarized as ontogeny recapitulates phylogeny. Ontogeny is the life history of an organism, while phylogeny is the evolutionary history of the race of that organism. The recapitulation theory not only supports the concept of organic evolution but also discloses the stages in the evolution of an animal. Recapitulation is seen in invertebrates also. Example, adult insects have 3 pairs of walking legs, but in embryo each body segment has 1 pair of primordia of legs. This shows that insects have evolved from an ancestor having segmentally arranged appendages. Certain animals show complicated larval forms and sample adults, example ascidians, parasitic copepods, parasites like Ciculina in retrogressive metamorphosis. Due to parasitic habit, adult organs are highly reduced. In some animals, example axolotl, larva of ambystoma, the larva fails to undergo metamorphosis. It develops gonads, attains sexual maturity, and starts reproduction. This is called neoteny or pedogenesis. Retention of primitive or larval features provides evidence in favor of evolution that under specifically favorable circumstances, natural selection favors retention of primitive or larval characteristics. Ecogeographical rules, size, shape, and color. Cope's law. In the course of evolution, there is tendency in animals to increase its size. Example, dinosaurs. Bergman's law. Warm-blooded animals are large in size in the colder regions as compared to hotter parts. Allen's law. Extremities like tails and ears become smaller in colder areas. Glauber's rule. Warm-blooded animals have more melanin in hot, wet areas but develop yellow-red pigment in hot, dry areas. Rappaport's rule. Species adapted to cooler climates have a wider range of latitudinal distribution than the species adapted to warmer climates. Jordan's rule. Fishes of colder waters have larger sizes with more vertebrae than the fishes of warmer waters. Dallow's law states that evolution is irreversible, proposed by L. Dallow in 1893. Gauss's law is Gauss 1934, or the competitive exclusion principle of Hardy in 1960. It states that the two species having the same ecological requirements cannot continue to occupy indefinitely the same habitat. Paleontological evidences. Paleontology is a study of the past life based on fossil records. The study reveals the existence of life in past and illustrates the course of evolution. The organic remains like bones, hairs, nails, claws, shells, or impressions of the organisms found in rocks are called fossils. Okay, on to the next page. Father of paleontology is Italian Leonardo da Vinci, 1542 to 1519. While founder of modern paleontology is Cuvier, 1769 to 1832. I think there's a problem with the date there, 1542 to 15. It should be the other way around, 1590 to 1542. Anyways, we'll check that later. Just verify it for yourself. Fossils are remains of extinct organisms buried and preserved by natural process. The science of paleontology links biology with geology and is concerned with finding, cataloging, and interpreting evidences. The study of fossil plants is called paleobotany, while the study of fossil animals is called paleozoology. Charles Darwin was the first to show that fossils provide direct evidence of organic evolution because it deals with the actual organism which lived in the past. The different methods of fossilization, a continuous process, are intact preservation, petrification, molds and casts, impressions, mummies, tracks and trails. The media in which fossils occur include sedimentary rocks, amber, asphalt, volcanic ash, ice, peat bogs, sand and mud. Age of fossils is determined by dating the rocks in which fossils occurs. We're talking about radiocarbon dating, right? The dating of rocks are called geochronology, and dating system is called clocks of rocks. Rocks are of three types: sedimentary rocks formed by gradual settling down of fragments in regions such as lakes or sea, example conglomerates, sandstone, shale, lime, gypsum, etc. Igneous rocks formed by the cooling down and solidification of molten material of the earth. Metamorphic rocks are actually sedimentary rocks which are changed by heat and pressure, example schist, gneiss, slate and marble. The method for determining the age of rocks or of the fossils are carbon dating method discovered by W. F. Libby, radioactive clock method, uranium lead method, which is based upon the disintegrating property of radioactive elements proposed by Boltwood in 1907, and potassium argon method. Half-life period of carbon-14 is about 5600 years. The common radioactive elements which lose the radioactivity and change into their non-radioactive isotopes at a fixed rate are potassium 40 to argon 40. Right, we're just trying to figure out the difference or any discrepancies, if at all any. right? And carbon 14 to nitrogen 14, uranium 238 to lead 232, not 7, rubidium 87 to strontium 87, thorium 232 to lead 206. The half-life of K40, potassium, is 1.3 into 10 raised to 9 years. Right? So potassium argon method is useful because potassium is a common element found in all sorts of rocks. Potassium decays into argon extremely slowly. Right? Remember this. Potassium argon method has recently been used to determine age of hominid fossils in East Africa. Living fossils are those plants and animals which have become extinct excepting one or two representatives: Sphenodon, ginkgo, equisetum, etc. Death and disappearance, extinction of large groups of plants and animals over a short span of time is called mass extinction. Mass extinction may be due to the crash of a meteor or comet with Earth impact theory, or to the drifting, coalescing, and breaking apart of continents, continent moving theory. Mass extinctions shape the overall pattern of macroevolution. Right? And the largest mass extinction came at the end of the Permian, about 250 million years ago. This coincides with the formation of Pangaea II, when all the world's continents were brought together by plate tectonics. The most well-known extinction occurred at the boundary between the Cretaceous and Tertiary periods. This is called the KT boundary and is dated at around 65 million years ago. This extinction eradicated the dinosaurs. Fossil forests, parks are large-sized exposures of fossil-bearing rocks dug out and conserved by experts so as to give glimpses of what sorts of plants and animals existed in that period. Right? And coming to the next point. The fossil forest parks are large-sized exposures of fossil-bearing rocks dug out and conserved by experts so as to give glimpses of what sorts of plants and animals existed in that period. Birbal Sahni Institute of Paleobotany in Lucknow is exclusively engaged in the study of fossil plants. Some national fossil parks are 1. 50 million years old fossil forest and Intertrapian lava sediments at Mandla and MP, Madhya Pradesh. 2. 100 million years old fossil forest in Raj Mahal Hills of Bihar. 3 is a 260 million year old coal forming forest of Orissa. 4th is a national fossil park. Tiruvakkarai is found in South Arcot district of Tamil Nadu. Fossilized trunks of trees were found. Of tree trunks, 20 million years old are kept in the Children's Park in Gindi, Chennai. A geological time scale. The geological time scale is a chronological order or history of evolution based upon the study of fossils. Geologists have divided the history of the Earth into eras, the periods, and the epochs. The Precambrian is a very long period of time. It comprises about 87%, the geological time scale. Paleozoic and Mesozoic eras are subdivided into periods, but Cenozoic is divided into periods and epochs. During Ordovician, the vertebrates originated. The first fish was Ostracoderm. First reptiles appeared during Carboniferous period of Paleozoic era from stem reptiles called cotylosaurs, the ancestral genus being Samoria. North America was the main center of dinosaur evolution. Now let's just go back to this table. It's a flowchart. All right, I'm just trying to read through that. The types of fossils, their formation and examples. Entire organism frozen into ice during glaciation. Woolly mammoths found in Siberia and permafrost 25,000 years ago. Entire organism encased in the hardened resin amber of coniferous trees. Insect exoskeletons found in Oligocene rocks in Baltic coast. The entire organism encased in tar, mummies of mammals and birds found in asphalt lakes of California. Entire organisms trapped in acidic bogs. Lack of bacterial and fungal activity prevents total decomposition. Mummies found in rocks and peat in Scandinavia. Hard skeletal materials trapped by sedimentary sand and clay which form sedimentary rocks, example limestone, sandstone, and silt. Bones, shells, and teeth very common in British Isles. Molds and casts, hard materials trapped as above, sediments hardened to rock. The skeleton is also leaving its impression as a mold of the organism. This can be infilled with fine materials which harden to form a cast. Great detail is thus preserved. Gasteropods from Portland stone, Jurassic casts of giant horsetails, calamites of Carboniferous forest, internal casts of mollusk shells showing muscle attachment points, fossils of Pompeii city which was hurried by volcanic ash, from Mount Vesuvius in AD 79, presents molds and cast of men and domestic animals. Petrification, gradual replacement by water-carried minerals, deposits such as silica, pyrites, calcium carbonate or carbons, slow infilling as organs decomposes, producing fine details. Silica replacements of the echinoderm microstra. Impressions of remains of organisms and fine-grained sediments on which they died. Feathers of Archaeopteryx in Upper Jurassic. Jellyfish in Cambrian found in British Columbia. Carboniferous leaf impressions. Imprints. Footprints, trails, tracks, and tunnels of various dinosaurs. Of various organisms made in mud are rapidly baked and filled in with sand and covered by further sediments. Dinosaur footprints and tail scrapings indicate size and posture of organisms. Coprolites, fecal pellets prevented from decomposing, later compressed in sedimentary rock, often contain evidence of food eaten, example teeth and scales. Cenozoic mammalian remains. Right. Moving on to the next page. First indication of fishes appeared in Cambrian period. In Devonian, diversification of fishes and evolution of amphibia occurred. The large Devonian is characterized by the appearance of the first land vertebrates. These animals were called stegocephalians. In Pennsylvania, amphibians became dominant on land and appearance of first reptiles occurred. In Triassic, transition of reptiles and mammals occurred. In Jurassic period, there were dominance of dinosaur on Earth. First toothed bird appeared in Jurassic period. Toothed bird, as well as dinosaur, became extinct in Cretaceous period. Evolutionary explosion of mammals occurred in Paleocene period. First man-like apes appeared in Miocene. In Pleistocene, extinction of many large mammals occurred and also referred to as Age of Man. Recent epoch, the period of the last 10,000 years of Earth's history, is the Age of Man, where the development of human cultures occurred. Elephants of Pleistocene period are commonly known as woolly mammoths. Elephants have originated from some common Proboscidean Sirenian ancestor. True elephants belonging to the genus Loxodonta and elephants appeared in Pliocene. True camels belonging to the genera Camelus appeared in the latter portion of Cenozoic era. Mesozoic era is known as era of intermediate life and age of reptiles or dinosaurs. The phylogeny of horse has been traced from the fossils discovered from the tertiary rocks of North America. The first known horse-like animal which forms the starting point of equine evolution is Hyracotherium, also called Eohippus. It is commonly known as dawn horse. Merychippus, ruminant horse, in Upper Miocene marked the completion of transition from browsing to grazing. Modern horse equina is the direct descendant of Pliohippus, if I got that right. Yeah, it's Pliohippus. First representative of equids appeared in a late Pliocene and during Pleistocene achieved worldwide distribution. I'll read up the details of the evolutionary sequence of horse. Eohippus to Mesohippus, that's Eocene period to Oligocene period. Then with the Merychippus, Miocene period to Pliohippus, Pliocene period. Then into the Equus was the Pliocene period. We have the recent epoch, a gene of age of man and limbs, Holocene, Pliocene, age of mammals, birds, and angiosperms, Cretaceous, age of reptiles and gymnosperms, Jurassic, period of dinosaurs, Permian, age of amphibians and ferns, Devonian, age of fish, Silurian, age of higher invertebrates. Biogeographical evidences. Biogeography is a study of distribution of animals and plants in different parts of the earth. A.R. Wallace, an English naturalist, divided Earth into 6 biogeographical realms: Palearctic, Oriental, Ethiopian, Australian, Nearctic, and Neotropical. Wallace line is an imaginary line between Oriental and Australian realms. 2 zoogeographical regions separated by high mountain ranges are Palearctic and Oriental. Prototherians, the most primitive mammals which provided evidence of organic evolution from geographic distribution, are found in Australia. Charles Darwin, during his voyage around the world, studied the fauna and flora of Galapagos Islands off the west coast of South America. Galapagos Islands are called a living laboratory of evolution. Darwin studied 20 related varieties of the bird belonging to family Geospicidae, which are different in shape and size of beak. These birds are now called Darwinian finches. Some years after Wallace's line, Weber drew an imaginary line on the map between the Oriental and Australian region because it was considered to divide the fauna of 2 regions better than Wallace's line. The Weber's line is further east than Wallace's line. Wallacea is a transitional area between Wallace's line and Weber's line in which one of the animals of both the regions are found. Thus the islands, that is Celebes and Flores and Lombok, lying between the 2 lines are neither the geologically part of the Oriental region nor part of Australian region. Biogeographical evidences are based on discontinuous distribution. Descendants of a common ancestor inhabit different continents, some mutated and slightly different but closely related species of the same genus, example alligator, Mangolikus, tulip, sassafras, etc. Restricted distribution. The continent has peculiar flora and fauna, example pouched animals in Australia, life on oceanic island. The animals and plants of oceanic islands resemble those of mainland, yet include distinct species, example Darwin finches of Galapagos Islands and the large-sized iguana and tortoises. Convergent or parallel evolution. In this natural selection can induce animals of 2 different groups to adapt similar habits for survival, example marsupials. Physiological or biochemical evidences compared to biochemistry also shows a relationship among the animals. The aspects of biochemistry that indicate biochemical affinity are metabolic processes, enzymes, hormones, blood and lymph, blood protein, blood groups, and molecular homology. The metabolic processes such as digestion, biosynthesis, respiration, muscle contraction, and transmission of nerve impulses occur almost in a similar manner in all animals. The enzymes are essentially alike in their nature and action in most animals. For example, trypsin is found in many animals from sponges to man. This shows that all animals are related to one another. The hormone resemble in their chemical nature and function in all the vertebrates. Thyroxine plays a role in the metamorphosis of tadpole into a frog. If the thyroid glands are removed, the tadpole fails to turn to a frog. Such a larva starts metamorphosis if fed on thyroid gland of another animal. Let me repeat that. Such a larva starts metamorphosis if fed on thyroid gland of another mammal. The blood and lymph fluids are similar in their composition and physiological role in all vertebrates. This indicates relationship among vertebrates. The blood proteins of various mammals are similar to a large extent, but sharply distinguishable from those of other vertebrates. The blood protein tests have shown that man is nearest the great apes, gorilla, and chimpanzees, and next nearest in order are the Old World monkeys and the New World monkeys and the tarsiers. The same is indicated by the anatomical facts. Thus, the blood protein tests reveal man's genetic relationships. 4 blood groups AB, AB, and O occur in humans. The blood groups A and B are found in apes but not in monkeys. This suggests that humans are beings are more closely related to apes than to monkeys. Though all the three have a common ancestry, the same is shown by blood proteins tests mentioned earlier. The occurrence of similar biochemicals in animals is called biochemical homology, just as anatomical homology is shown by similarity in organs and tissues. Similarity among animals at the molecular level is called molecular homology. Human DNA differs in only 1.8% of its base pairs from chimpanzees' DNA, and there is no difference between the two in the amino acid sequence for the protein cytochrome C. Close relationship indicated by these sequences conforms to show that by other means, such as anatomical or fossil evidence, physiology of excretion in vertebrates clearly indicates that they have been evolved gradually from simple forms to complex ones. The nitrogen as waste in many fishes, example bony fishes, is in the form of ammonia. In case of amphibia, it is urea, as amphibia are more evolved and adapted for life both in water and on land. Taxonomical evidences. Taxonomy is the study of the theory, practice, and rules of classification of living and extinct organisms. The naming, description, and classification of a given organism draws an evidence from a number of fields like classical taxonomy, based on morphology and anatomy. Cytotaxonomy compares the size, shape, and number of chromosomes of different organisms. And numerical taxonomy uses mathematical procedures to assess similarities and differences and establish taxonomy groups. In the present system of classification, different systems, labels, or taxons are employed, like species, genus, family, order, class, phylum, etc. Different species possess similar features are included in a genus. Several genera with certain common characteristics are found grouped into a family. Similar families form orders, classes, and phyla. The classification further shows the grading of different phyla in the order of increased complexity of organization. As classification reveals a positive relationship between different animals, which is due to their descent from a common stock. Evidence from genetics. Genetics is the study of heredity, the transmission of parent characters to the offspring. A number of sudden inheritable variations are called mutations. In mutation, new genetic material is created which never existed before. Mutations provide raw material for evolution and can give rise to new species. Heredity variations also occur due to genetic recombination. The latter causes hybridization. Examples of hybridization mutations are available, which shows that evolution has taken place. Pattern of transmission of characters is also similar in various organisms. It shows interrelationships among different living beings. Man has developed many variations of useful animals, example cows, ankon, sheep, horses, hornless cattle, etc., and crops, example wheat, rice, cotton, etc. Some variations of fruits, like mango, citrus, etc., have also been improved. Hybridization and induction of polyploidy has given rise to new plants like Triticale, Brassica. Such studies have provided useful evidence concerning the evolution. And that is the end of this chapter. Thank you for listening, and we'll discuss details on Q&A in the next lecture. Thank you. Have a great day. And do please make notes while you read and understand them well. Any questions, put them in the comments section. All right? Okay, have a nice day.